A-Level Biology 9700, Paper 4, Variant 2, October, November 2025. And in this video, we will discuss question number 1 and question number 2. So question number start with a flow diagram in which rice, Oryza sativa, is a crop plant that has adopted to grow in flooded fields. Figure 1.1 outlines the effects on rice plants of growing in flooded fields. So the flooded fields, lower concentration of mineral ions and capital A in the soil available for absorption compared to non-flooded fields. Three arrows arise from this one statement. The high concentration of waste product B in the root cells. The second arrow shows the presence of tissue C in stem and roots. And the arrow 3, production of ethene. So the third arrow also arising one arrow breakdown of cyclic acid ABA. So with reference to figure 1.1, name these three: substance A, substance B, and tissue C. So first of all, this one A, B, and C. We have to write their names. So as you know that we have to identify these. So in flooded fields, in the flooded fields, water replaces air spaces in the soil, which leads to the low oxygen conditions. So air will be the A will be the oxygen. And second, high concentration of waste product B in the root cells. So due to the lack of oxygen, root cells must switch from aerobic respiration to alcoholic fermentation, which produces ethanol as a waste product. So B might be the ethanol. So the C is parenchyma. 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 So this is specialized tissue containing large air spaces that allow oxygen to diffuse from the aerial parts of the plant down to the submerged roots. So this is parenchyma. So I'm going to write all these on the given space. So on the given space, A substance is oxygen. B is ethanol. And the C is parenchyma. Now, part B of this question is the describe how root cells respond to a high concentration of substance B. Substance B, ethanol, is produced as a result of a shortage of oxygen. Ethanol is produced. So root cells increase in the production of an enzyme which is called as ethanol dehydrogenase. So this enzyme helps break down ethanol through the primary goal is to keep the glycolysis pathway running. So response is the formation of or the production of an enzyme ethanol dehydrogenase is increased. To break down ethanol. Next question is gibberellin is involved in the growth of stems in rice plants. Figure 1.2 shows the effect of gibberellin concentration on the length of stem in the rice plants. So here you can see that a graph which is figure 1.2. On x-axis, they have taken the concentration of gibberellin in arbitrary units, and on the y-axis, they have taken the length of the stem in millimeters. So here you can see the graph starts from the concentration of gibberellin starts with 1, and whereas the length starts from 0. So you can see as the concentration of gibberellin is increasing, the length of stem is also increasing. So they have, you can say they are directly proportional to each other. You can see that the concentration of gibberellin and the length of the stem, they are uh, directly proportional to each other. So I would like to say that there is a positive correlation. There is a positive correlation between the concentration of gibberellin and the length of the stem. As I have told you that the concentration of gibberellin is increasing as the stem length also is increasing. So here I would like to say that if we add some values from the graph, then I would like to say that at a concentration of one arbitrary unit, uh, the stem length is 4 millimeter. Whereas at a concentration of 5, you can see at a concentration of 5, you can see the length has been increased to 52. So the increase is not perfectly linear, but the steeper increase occurs between the concentrations of 3 and 4 arbitrary units. So the question has 2 marks. So according to 2 marks, I would like to write as much as possible, but you have to choose accordingly. So I would like to start with there is a positive correlation between concentration of and the length of stem. As the concentration of gibberellin is increasing, stem length also increases. As at concentration 1 of gibberellin, the stem length is 4 mm, but at concentration 5, the stem length becomes increases as 52 mm. Next part of this question is ABA in rice plant cells inhibits the action of gibberellin. With reference to figure 1.1 and 1.2, explain the importance of ethylene production for the growth of the rice plants in flooded fields. So actually this question is connecting the inhibitory effect of abscisic acid ABA with the stimulatory effect of the gibberellin and the role of ethylene. So I would like to write some points which will lead to the answer as ABA, or you can see ethylene acts as a signal to decrease the concentration of ABA in the cells. And rice plants are submerged in flooded fields. They face low oxygen levels, which triggers the production and accumulation of ethylene gas within the plant tissues. So the prompt states that ABA inhibits the action of gibberellin. The reduction of ABA allows gibberellin to become more active or increases in concentration. So as a result, you can show in figure 1.2, the higher concentration of gibberellin leads to rapid stem elongation. So this rapid growth allows the leaves and flowers of the rice plant to stay above the water level. This is crucial for maintaining gas exchange for the process of photosynthesis and aerobic respiration and ensuring successful pollination. So these are the details. So you have to go for 3 marks. So you will start as when rice plants are submerged in flooded fields, they face low oxygen levels or concentration, which triggers the production of ethylene, as we had discussed in the question 1, which acts as a signal. To decrease the concentration of ABA. The reduction of ABA allows to become more active, which leads to stem elongation, stem elongation above the water level. for gaseous exchange. So it was all about question number 1. Now moving towards the question number 2. Question number 2 is explain the terms gene, genotype, and phenotype. Very simple question. Directly they have asked about the definitions. And gene is the um, a small segment of DNA, or we can say that a length of DNA that codes for a specific polypeptide or protein. So it is the basic unit of inheritance located at the specific position or locus on a chromosome. Then second, the genotype is the genetic makeup of the organisms, and phenotype is the observable physical characteristics or features of an organism resulting from the interaction between its genotype and the environment. So we have to write these 3 definitions for 1 mark, 1 definition for 1 mark. So the genetic makeup of an organism in terms of it was to be right on the lower one. And second, the genetic makeup of an organism. Phenotype is the physical appearance the physical or observe the physical observable appearance or characteristics So these are the 3 definitions. You can also write one example for each. Now the second part of this question is a monohybrid genetic cross can be carried out to produce an F1 and F2 generations. Outline how a monohybrid genetic cross is carried out. State the expected percentage of each of the different offspring genotypes in the F2 generation. And here students assume that the inheritance pattern is for autosomal dominant and recessive alleles. Well, monohybrid cross actually explains the law of segregation, and it was presented by Mendel, and for this purpose the parental generations are needed. That is, the two pure breeding homozygous individuals with contrasting traits are crossed together. As a result, that parental generation, the cross between those parental generations, the F1 generations, the first filial generation is produced, in which the all offsprings are heterozygous and they display the dominant phenotype. And then F2 generation is uh, can be reproduced as which allow the F1 individual to self-pollinate or cross with one another. So as a result, the expected rate, percentage of the genotype F in F2 is changed. So in an autosomal dominant recessive pattern, a cross between two F1 heterozygous yields the following genotype ratio, that is the 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. So I would like to write all this in a very not very short, as the question is of 5 marks. So I would like to start with the monohybrid cross, or we can say that a monohybrid cross between two homozygous parents with contrasting traits. Homozygous means capital A, small a. One will show the dominant characteristics, the other will show the recessive characteristics. So the capital letter shows the dominance, and small letters will show the recessive characteristics. So as a result of their cross, F1 or first filial generation is produced, in which all individuals will be heterozygous. Heterozygous as and display dominant trait or phenotype. So then allow the F1 individuals or the organisms to self-pollinate to get F2 or second filial generation. So in an autosomal dominant or recessive pattern, this grow this cross results the following genotypic Roshan. Ratio. Homozygous dominant. %25. Heterozygous, capital A small a, will be 50%, and homozygous recessive, small a will be 25%. Or in other words, we can say that the ratio can be 3:1 because heterozygous condition is also displaying the dominant trait. So the ratio can be 3:1. So it was all about question number 1 and 2. Please like, share, and subscribe my channel to watch more videos on this paper and other papers as well. Thanks for watching.